This week in lab, we'll be starting our RT-PCR section, or section 1, where we analyze the differential effects of our environmental toxin on gene expression, right? Genes of our choosing. So just as a reminder, or an outline for this short pre-lab lecture, I'm going to remind everyone about the utility of RT-PCR, right? What does it measure? How does it measure that? Very briefly, if you need to familiarize yourself with the topic, you can review the RT-PCR overview at large. And the goal of today's or this week's lab is to design some primers to investigate the expression profiles of genes of interest. And similarly, I will highlight their role, again, the key step they play in reverse transcriptase PCR. And then I will briefly touch on the project requirements and or the things you need to accomplish for this week, and as well as the assignments you need to upload. So as a reminder, RT-PCR is a quantitative or semi-quantitative method that biochemists and cellular and molecular biologists use to quantify gene expression. So this tool allows us to measure the mRNA changes or the changes in mRNA transcription, and we can compare them to compare their relative expression profiles to a housekeeping gene. So ultimately, this allows us to measure whether genes are being transcribed more or less, or perhaps not changed at all, right? It literally lets us measure quantitatively or semi-quantitatively the effects our environmental toxin will have on gene expression, right? Whether that drug or toxin makes it go up or down. The first step of which we have to choose our genes, which is what we're going to be doing this week in lab. And just as again, another reminder that this is the section 1 of the lab section, or excuse me, section 1 of our lab course, and this step investigates that transcription step between DNA, right, where genes are stored in our nucleus and our genome, and then transcribed into mRNA. So that's section 1, or the first portion of the central dogma of biology. Again, if you aren't familiar with this, just review the central dogma of biology video at large, as it's rather lengthy. So this week, again, we'll be designing our own primer sequences that will allow us to investigate genes of interest, right? So genes that we're interested in measuring whether their expression goes up, down, or doesn't change at all. So in order to do this, we first have to design our own primers. And where we're going to do that will come up in a few slides. But the primers that we're designing, just to reemphasize and bring back to where they play a role in our RT-PCR process. So we've discussed previously in the PCR overview video that as we place our reactions or our samples into our thermocycler, which is going to heat up, cool down, each cycle of that heating will help break apart the 2 strands of DNA molecules that are produced. And in order to selectively prime the amplification of our genes of interest, we need to artificially produce primers or primer sequences that are selective for our genes of interest, right? So if you've watched the video, the primers that we'll be designing are these guys right here, right? So we're going to design a forward and a reverse primer sequence. You need both in order to amplify both strands or copy both strands of our synthesized DNA products. And these guys literally serve, again, to prime that DNA polymerase as it copies our DNA segments over and over and over. Right? So we're literally designing sequences of nucleotides that will prime amplification of our genes of interest. And just to reemphasize, we need to design these specifically for our genes of interest because we don't want to amplify other genes, right? And to add another cartoon, just in case you've been looking at this cartoon infographic, you can see primers right here, right in green. So here's the first or second step in PCR, or the first or second cycle, and then as you can see, these primers prime the synthesis of new DNA strands. Another cycle repeats, more primers come in, more DNA is synthesized, and this process repeats over and over and over. So these little green guys here are what we're designing and utilizing. Well, we're designing them this week. We'll utilize them in a few weeks. So the first step in doing all of this, designing our own unique artificial primers, is to pick a gene. So you and your group will work on this separately. You guys need to decide, you need to decide on your tissue or your process or your signaling cascade, right? You need to start with a topic of your choice. After that, you need to decide and choose which specific genes you'll be analyzing. After that, if you follow this week's protocol, you will end up on the NCBI database, where there are a slew of just a lot of different biochemical, cellular, molecular information. If you see in this pull-down tab here, you can look up things like sequences and the role or jobs of specific genes in various species. You can look up genomes, and this is technically cut off, but if you were to go further down, you can also see things like different proteins, the amino acid sequences, expression profiles of different proteins in different species. There's just a vast amount of information stored on the NCBI website. The way we're going to use this today, again, after you've chosen your gene, you're going to want to go to NCBI, the NCBI website, select the gene in the pull-down menu here, and then plug in your gene of choice. This is alcohol dehydrogenase, and you're going to also want to include the species that we're investigating this in, right? So in the lab, we're looking at zebrafish. So the scientific name of zebrafish is Danio rario, because we're only interested in looking up and comparing or analyzing the genetic sequences and gene expression of zebrafish, right? If you've been paying attention in class or you've been reading our textbook, you'll know that certain sequences of genomes are shared, or very shared in the sense that they're very similar between different species, but typically they're not identical. So we don't want to look up the genetic information or genetic expression profiles of alcohol dehydrogenase in, say, mice or rats, right? Make sure you are identifying the correct species when you start this process. And again, all these details are in our protocol, so just follow the protocol step by step and you won't get lost. So just to emphasize here, groups of 4 must choose and research 2 novel primer sets, right? So we've been supplied with a list of primers that are currently available and that have been used before. So depending on how many you have in your group, I'll touch on this in the last slide also, but depending on how many students you have in your group, you'll be required to pick 2 novel primers. I'll highlight that again on the last slide. And your job for today is to choose those genes, research and uncover / build those primer sequences, and then once you have specific information from the last step in this process, you're going to highlight it and upload it into the Excel sheet assignment on our Brightspace page, right? Download the Excel sheet, enter the specific information that you have to, and then upload the assignment so that I can compile things and buy the reagents that we need to. So this is a representative slide of the last step in today's protocol. Again, if you're picking 4 genes, you will have to do this 4 times. If you pick 3 genes, or if you have a group of 3 and you pick 3 genes, you will have to do this 3 times. But this is just a representative picture of the data output you should be arriving at today. So this is a screenshot of primary information to put into the Word document at the end of your protocol. And again, there is information here that you need to take and enter into your Excel sheet. So specifically down here, I have arrows pointing at the product length. So here's, we've gone through the whole protocol. Again, this won't make any sense unless you follow the protocol, so please do work your way through the protocol before you think that you can arrive here. So at the very end, again, there's information here in our primer pair 1, which is the primer pair we're going to choose. product length. So this looks like 340 here. It may be hard to see on the screen, but it's just this arrow points to the location of the product length. This is one thing you will have to report. In our Excel sheet, this is called amplicon size because product length essentially is the size or length of the DNA segments that we're amplifying. And then you will also have to report annealing temperatures. So over here, this arrow points to this column, TM, and it looks like this is roughly 59.9 and 60.04. So this is the annealing temperature for the forward primer, and this is the annealing temperature for the reverse primer. Remember to include and report these on our Excel sheet because this will affect which protocol and the specifics of which protocol we choose to use during our amplification reactions in the weeks to come. So here, just reminders and clarifications about the assignment. So each student must have at least one primer set, right? So groups of 2 should pick 2 genes, develop 2 primer sets. Groups of 3 should pick 3 genes to investigate and develop 3 primer sets, etc. 2 of these, if you're struggling to choose which genes or how many genes of which kind of target, 2 of these genes should at the very least be specific for your tissue or organ or signaling cascade, right? Whatever topic you've chosen to study the effects of ethanol on. And then if you're struggling to find genes that are specific to, say, if you picked cardiac tissue or a signaling pathway in cardiac tissue, and you are struggling to find more genes for that, you can also choose to include one general metabolism gene or an oxidative stress gene, right? Exposing our developing zebrafish larvae to our environmental toxin will undoubtedly cause changes in metabolism and changes in oxidative stress responses, for better or for worse. And maybe there's a way you can tie the results that you observe in these genes back into the effects of your other genes for your specific tissues or organs. And then last reminder, be sure to download the Excel file, include the specific information you need to, right? Annealing temperatures, amplicon size or product size, the specific primer sequences themselves, as well as gene name. Include those in that Excel file, and then be sure to upload it as an Excel file. I will not accept submissions that are submitted in PDF or Word or other things. If you have any questions, feel free to email me before lab.